Okay, folks, going to start. So the next speaker is Gestur, who is the PI at the Department of Experimental Immunohematology, and the head of immunoglobulin research at Sanquin Research, and a professor of immunoglobulin biotherapeutics at the Department of Pharmaceutical Sciences at Utrecht. So Gestur studies humoral immune responses, FCRs, complement, and particularly the role of glycosylation in these responses. So, Gestur.
Okay, thank you very much for the introduction and invitation to this wonderful place in a boat. I'm happy that we need not on open seas and that you remain your normal complexity. And me too. So I first have to explain a little bit from where perhaps I am. I'm indeed from Sanquin Research, which is a not-for-profit organization involved in basically blood transfusion medicine. But we also have a research unit where we study basically human immunology in that context. And we often look at anti-red blood cells and antiplatelet responses as a learning tool to study human antibodies, at least in my group. And we're kind of used to know what these antibody receptors do on myeloid cells and NK cells in terms of different subclasses of antibodies. We also know what they do in terms of complement, and you can sort of pick your flavor here in terms of antibody subclasses. But I'm going to talk about something different, which is the variability in the Fc region on glycosylation. And I'm going to try to convince you that there is a sort of a parallel universe of those subclasses out there with different glycosylation and different function. And I'm going to go through what these switches are, how they are turned on and off, and basically what they mean for human health and diseases. So basically, well, we all know that's the antibody structure, the diversity in the variable region, but we more or less have not really embraced the variability in the Fc region, which is obviously not constant. And the reason why I actually started looking at this region in particular for immunity to red blood cells and platelets is that very often you have these responses in pregnancy where the maternal immunity starts to attack the fetus red cells and platelets, and they become anemic or thrombocytopenic, which can be a very life-threatening disease in and after pregnancy. And there the antibody level and the titer thereof is not really predictive of disease, and avidity and affinity also not, and subclasses also not. So we started looking in regions that are not really there. And as I mentioned, it's highly diverse. So if you look into, this is a glycopeptide analysis of the Fc region of IgG1. And basically what you can appreciate is that it's quite diverse. So you have a basic core structure, which is consisting of N-acetylglucosamines and mannose. But it can be further decorated by bisect and GlUGNAC, 1 or 2 galactoses, and on top of that, 1 or 2 sialic acids, and a fucose. And I think we all knew that, and probably most of you know this, that it is there and it's variable and it has something to do with the antibody function, but also that afucosylated antibodies are something, a therapeutic modality to increase affinity to Fc gamma receptor 3, more like a scientific curiosity that sort of popped up at some moment because of a cell line that produced afucosylated antibodies. Afucosylated antibodies are almost not present in our blood, and if you take a look at these small peaks here that I've highlighted in red, they are a minority in our blood. So those are the structures here without the red triangle, which is the fucose, which I will be spending a lot of time on in a couple of next minutes with in the first part of my talk. So basically it's not there, so why would that be relevant for our human immunity? Well, first of all, of course, is that this fucus is not on the outside, it's on the inside. It's a really confined space that is not really easily accessible. But I would like to sort of draw the attention that even though it's only a very small minor fraction, of the total blood, it really matters the context what you're looking at. So because although fucosylated antibodies, the normal situation that we have, afucosylated antibodies is really found as the majority of antibodies you find in some immune responses, and I'm going to highlight which responses these are. So it started at this alloimmune pregnancy situation, where and now I'm just showing the anti-HBA1 responses, that's human platelet antigen 1A. It would be identical if you would look to anti-rhesus antibodies, red cell antigen. So and each dot here is just a typical picture of this, is one single maternal blood, and you look at total IgG, it's just fully fucosylated, almost 94% on average, which is normal. But if you look at the corresponding specific antibodies, they are from also highly fucosylated down to almost afucosylated antibodies. So it's really diverse, almost a normal distribution. And the question was, what does it do? But perhaps also very curious observation is that when you look in time, these are extremely stable structures. So even if you immunize somebody here and through pregnancies, so these are pregnancies up to 11 pregnancies, you see that when we have a sample in early time point, so in the first pregnancy, it seems to start sometimes high and then they sometimes drop, and then it seems to stabilize and be stable in very long time. And we see that also in blood transfusion up to 10 years in time, when you set a certain immune response in early in the response, and then just you stay with that fucosylation level for apparently for at least 10 years. So it's there, but there are also other structural changes in the glycan. So this is fucosylation, and bisection is very similar thing. It's more diverse, but calculation is also quite diverse. And cellulation, which is normally not there, is in the early responses in pregnancy also elevated, and certainly in the specific antibodies. And I will also mention what these glycan features do, but I will focus on this fucosylation in the first part of the talk. And so to study this, we really simply started with HEK cells, and we basically used these simple tools to produce our antibodies, as most of you also do, I think, but I included also substrates and enzymes and co-transfected them with enzymes in the whole machinery to actually attach these glycans or block those incorporations. And in this way, we could generate basically quite efficiently and reproducibly antibodies of any desired glycoform with either fully fucosylated, and then we can take that down to 20% or a little bit higher sometimes, with or without bisecting glucnag, increasing calcylation and cellulation. So basically in context of each other. And using this sort of a setup, we set up also in conjunction an array with all the Fc receptor, where we see terminally a couple of biotin tag on there, and then we different concentration, and we flow each of these variants thereover, and we can assess the affinity basically. And to make a long story short, for all the Fc receptors, Fc gamma receptor 1, 2, and 3, it really doesn't matter what kind of glycoform you have, at least under these circumstances, it doesn't matter. The affinity is not changed. But for Fc gamma receptor 3, we know that it's expressed on NK cells, the only receptor on NK cells, and highly expressed on some macrophages and monocytes, is that when you take off the fucose, you get a really bump into the affinity, which is 20-fold, or up to 40-fold if you also increase the calcylation. And cellulation has something to do with it, but not really much. And bisection really doesn't do anything. So basically, these are bisected, non-bisected. And again, this pattern is repeated over here. But basically, the fucose does all the job here. And the question is then, was always then, why is that happening? And the reason is that if you look into the structure, Fc gamma receptor 3, and only Fc gamma receptor 3 family has a glycan on its own. The other receptor don't have this glycan that sticks into the Fc region, and basically is blocked by this fucose if it's there. And if this fucose, if you take it off, this glycan can be much better accommodated for, and therefore you get this bump in affinity. I think that's quite established now. But what is even more striking is that when you look at NK cell activity, is that basically, and this is not fold activity anymore, this is just activity, it's just zero for the fucosylated antibodies. Take off the fucose, the antibodies get function basically, and this is ADCC against red blood cell lysis by NK cells. And it's also modified by galactosylation and the other glycans in very similar fashion as we saw for affinity. And this is not just a snapshot because of concentration. And so we did this also for other antigens like the anti-K antigen. We make recombinant antibodies and we block the incorporation of fucose or leave it on. There is a black and white effect. And if you increase the concentration-dependent NK cell activities, it's just flatline for those fucosylated antibodies. Afucosylated antibodies, you get this bump in activity as well, which is really black and white difference. And we think that this is because you have, of course, this multitude of affinity interactions are going on, but that translates into avidity interactions. And these cells, of course, the effector and the target is both negatively charged. You need to really sort of accommodate that these guys meet and start to, attack each other, at least from the NK cell side. So, and if you go into the patients, so back to the pregnant women and the fetuses thereof. So basically you see these really huge differences in fucosylation again in the specific antibodies, not the total. Calculation is extremely increased as well. I'm not showing the other features because functionally they don't really mean much, as I will continue showing. And if you look at the level of calculation, does not seem to predict NK cell mediated lysis. Fucosylation seems to do so, and if you look at the hemoglobin levels in the fetuses, it also seems to be correlating with the amount of fucose. So the lower the fucose, the less hemoglobin levels, the more lysis you are seeing basically in the children, and the titer is not really giving a good prediction. So what we think is that it's not the quantity of the antibody, but it's the quality of the antibody that really matters in these diseases, and it has bearing basically to diagnostics, but also, of course, for therapeutic applicability. But you know more about that as well. So basically, until now, I've been telling you this afucosylated response is really sort of something associated with severe disease in autoimmune-mediated diseases. So in pregnancy, something that is really evolutionary conserved. We need to be reproductive to continue our species. And then the question is, these are really unique responses, and why are we making this? We know now that you don't need to be pregnant to make them, because you can also immunize men with rhesus positive blood, so RHD negative men also produce afucosylated antibodies just like females. If you transfuse platelets also to people with HLA mismatch in need of platelets, they also make anti-HLA antibodies that can be afucosylated. Same thing to anti-HLA antibodies in kidney transplantation, as Margie Ackerman has shown very recently, very similar to us. But then in kidney transplantation, the more severe the afucosylation is, the more chance is that you have a rejection of your graft, basically. So the question is, why are we making these crazy antibodies in pregnancy? Something that we should really precious for our species. It should not be happening, and not against the most infectious disease that we know of. We know that not all rhesus antigens, also red blood cell antigen, are capable of doing this. The T antigen is really good at it, very clinically relevant. K antigen is very, very clinically relevant, also doing this sometimes, but not the C and the E, and these are considered not clinically relevant antigen in transfusion medicine. So for some reason, some antigens do this and some others don't, and we don't really know that. But it's really seemed to be a crucial common element that these are sort of a cell surface membrane antigens, like other people have seen in HIV. But Galit Alter, but also Jeffrey Ravitz's group also found this in dengue, that is associated with, so in HIV, Galit and Ackerman showed that, since suggested that elite controllers seem to have more tendency to have these afucosylated antibodies. That can make sense, that they have better ADCC potential. And dengue, it seems to be the other way around, the more pathology you get. And that also makes sense if you have antibodies that promote the disease pathology. So then we question ourselves, so if you look at normal responses, they're all fucosylated, almost fully fucosylated antibodies, wherever we looked at specific responses, like soluble proteins, polysaccharides, it's all fucosylated. And then we started to look at, is it something specific to enveloped viruses and other proteins that are found on the surface of cells that we make these kind of responses. And we started comparing different antigens like anti-CMV, HIV. We also see the specific responses can be afucosylated. It really matters what kind of response you're looking at. It does not seem to happen, at least in this non-enveloped virus, to non-enveloped viruses. We also see this in HPV, natural immune responses, but not if you're vaccinated against the same component that is a protein vaccine. So also in line with our hypothesis that you need something surface structure that can be be injected through and seen by our immune system in that context. You also see this by mumps and measles, but it's really also by live vaccination, so attenuated vaccine. But to a lesser degree than these two viruses, anti-CMV, HIV seem to be really good at this. And then we looked at also SARS-CoV-2, and there, to our really big surprise, when we could look at this in a longitudinal way, is that almost everybody seemed to start with a slight afucosylation in the beginning, very early on at the seroconversion. It's much more pronounced in people with really severe disease. This is not only what we've seen, but also a group of Taiwan have seen this as well. So you start with afucosylated response, and then you fucose convert in time somehow. And we really don't understand that. I will come back to that later in terms of vaccination. I won't dwell on that. So basically, this is what we've seen. So enveloped viruses, so wherever we have looked at, seem to do this. They induced afucosylated responses. And I want to perhaps stress one thing is that because this is fucosylation level. Normally you have 100% fucosylation, and you could ask the question, so what does it matter, 20%, since 80%? But you have to think that these antibodies are the functional part of those antibodies. So it's going from 0% afucosylated antibodies down to 20. That's 20-fold increase, basically. So that's a huge, much huger number if you put it in this way. So then we asked ourselves the question, so what about other kinds of proteins that are also should be seen by the immune system, like provided by malaria and the red blood cell states, because these parasites, they hide within these red cells, they produce protein that actually you can see that they are sort of deforming the red cells. They need that to stick to endothelial cells, otherwise they're gobbled up by macrophages in the spleen. And then we looked at these two proteins that are expressed on those red cells, and you see indeed that here everybody in endemic regions seem to make afucosylated antibodies. And now it's not a fraction, or it's not going from 100 down to 10, but it's everybody make afucosylated antibodies, basically. And this is a pregnancy-associated malaria antigen. Also there, you seem to make this, and I'm not showing it here, but the number of pregnancy that these females undergo, where they start to make these antibodies, also correlates with the amount of afucosylation, which seems to be not happening to the merozoite state. That's the blood-borne state that is not associated with the red cells. So, and if you vaccinate somebody with the same antigen as a protein vaccine, again, you don't make afucosylated antibodies. You need the cellular context. It's 100%. So it's a really black and white difference in terms of that response. And a similar thing, and because of time, I'm not showing that, but ADC responses by NK cells is also here, black and white. If you afucosylate the antibodies, they have a good NK cell-mediated response. So then we asked ourselves, what about the vaccines that we're now introducing, the mRNA vaccines, the adenoviral vaccines? So basically, these are vaccine shuttles that actually give instructions to cells that produce the antigen, which is different from normal immune responses, and more or less should be similar to what we have been hypothesizing, that you should make an afucosylated response. And indeed, that's what you seem to see. This is basically antibody levels from naturally immunized individuals, ICU or mild infection. This is the antibody levels that you get. If you're naive vaccinated, you get really good titers, of course. And if you have been immunized before by natural infection, you do get a very strong response very quickly. And if you look at the afucosylation response or fucosylation response, ICU, as I told you, they are a little bit lower. than the mild, and the naive individual also seem to start here very early with some afucosylated response. Again, this is 100 down to up to 10. That is the afucosylated response. But again, here it's transient. It's going early in the response. You have afucosylated antibodies, and then they go down. But the curious thing is here is that those that actually saw SARS-CoV-2 before and they get immunization, they actually drop in this fucosylation level that seem to be more stable. And we're still looking into that, what that means in terms of boosting and boosting and boosting. We don't know that yet. But there is another issue here, and that's the calculation dimension that I mentioned to you before. It seemed to affect Fc receptor binding as well on top of afucosylation. But it does something else, and I'm going to show you that again. And this is a really curious thing, is that normally it's really sort of very high in this immunization. But what we saw in the ICU people is that they also start very high when they're sort of starting with the disease. They become infected, they make the antibodies seroconvert, really high galactosylation levels. Normally it's around 50%. This is extremely high. But these actually, only the very severe individuals, they drop really quickly in this calculation of those antibodies, and we still don't understand that. And this is something that we see also in total IgG. They start, so this is the antibodies completely not relevant to SARS-CoV-2, just what you find in blood. They're also going down. It seems to be something systematic happening under these inflammation or conditions that are changing not only the specific antibody, but also total antibodies. While the mild individuals, they completely are flatlined and nothing is happening there. So what is this calculation feature? Why is it important? Well, it's even more diverse than fucosylation. It's from, in any given age group, it's really variable. You can see that from males and females, it's very different. As we age, we are dropping our levels of galactosylation all the time. It's happening very quickly during menopause in females. And it has been associated with a lot of inflammatory diseases, autoimmune diseases. So almost everywhere where you looked at, you see that galactosylation is much lower, and therefore having an galactosylation profile in your plasma seem to have been sort of given the pro-inflammatory status of antibodies. But I think that's completely incorrect. On basis of what I'm going to show you now, and that's because when we make these antibodies, these glycoengineered antibodies, you characterize them in terms of fucosylation, bisection, etc. So these are basically 4 quadrants. On this side, it's fucosylated antibodies as before, and without fucose, bisected, non-bisected, and a non-bisected and bisected. And then in each quadrant, you increase the level of galactosylation, and then cellulation on top. And I think you can appreciate this pattern immediately, that C1q binding seems to have something to do with the level of galactosylation, but also the complement activity downstream C4 and down to lysis of red cells in this case. So the higher the level of galactosylation, you get stronger complement activation. And then the question is, how is galactosylation doing that? And to try to explain that, I have to go down to old literature of Dennis Burton, I think, from a nice review that you wrote a long time ago with Jenny Wolf, where you sort of suggested that you need this clustering to actually induce activation, and on surfaces this is happening. And then Paul Parent took over with his GEMAP colleagues and Pete Cross and produced these nice functional description of this hexameric structure that you need to get C1q binding. And then basically the question is how galactosylation inducing complement activity. Well, you could envision that it could either have something to do directly with the glycan interaction, C1q binding here to the Fc region, but that seems to be very unlikely given that the proximity is simply not there. It could be that the galactosylation is inducing changes in the CH2 conformation, so giving a higher affinity to C1q. Or it could be that the galactosylation features are changing the dynamics of the whole overall Fc structure, allowing it to form hexamers even better on surfaces. In order to test that, we went to, by Thijs van Os, using mutations described also by GEMAP, where we use the RGY mutations that form actually hexamers in solution, or hexameric mutants that actually give better rise to hexamers on surfaces of cells, or we use these charged residues that actually, when used alone, they actually sort of inhibit complement activation, or you can use them together and then they sort of complement each other. And then using these mutations, we hope to see that if you can pre-make complexes and see what calculation does, and if one of those hypotheses in terms of what calculation is doing may sort of present itself. And first of all these variants of antibodies that we made, they have nothing to do with antigen binding. They are basically binding the same, no matter what mutations we put in, no matter what kind of glycosylation we put in. And then we looked at complement activation, RGY, so wild type and the charged variants, they are here in the middle, basically. So C1q binding, C4, C3, and cell lysis. You see that the hexameric variants, RGY and the E430G, they are really good at it, as we knew and expected. If we now look at the glycoengineered variants, you see that the wild type variants are giving enhanced complement activation, as I showed you before, from C1q binding and trickling down to the lysis pathway. The charged variant, they do bind C1q and activate some C3 and C4, but they do not lyse the cells. Unless you actually combine them again, then they become very much similar to what you see in the wild type. But if you then put, but they all sort of, that's very important, have a benefit of having a galactose, but not so much sialic acid. So it's really primarily the galactosylation that is giving this enhanced complement activity. And it's really all down to C1q binding. If you then use these hexameric variants, either the RGY or the one that is promoting hexamer formation on surfaces of cells, all of them are basically flat lines in terms of what galactosylation does. So galactosylation does not do anything, or sialic acid doesn't do anything in terms of helping the antibodies or performing better. So what we think is happening is that galactose is not sort of enhancing affinity or changing the conformation in such a way that C1q has an intrinsic higher affinity to C1q, but it's promoting a better hexameric formation on the surfaces, which obviously RGY has no benefit of. And to test that even further, we looked at hexamer formation of the RGY variant in solution. So in solution, that's a concentration-dependent effect. So if you increase the concentration of your antibody, they are at low concentration, forming only monomer. If you make them sort of a higher concentration, they start to form hexamers. And if you now do this basically with, in terms of with the glycoengineered variants, you see that at lower concentration, this hexameric fraction is much higher or was relatively higher in the antibodies with enhanced galactosylation. Basically, indeed, suggesting that the galactosylation is somehow the principal component in normal antibody response that sort of can shift the balance of how these antibodies activate complement. And this basically is not only us, but a very similar time people at Genentech also showed very similar findings that galactosylation is doing. It's exactly in this way. And this is also happening in vivo. So we have also, because we are a blood bank, we also monitor this kind of transfusion with platelets, some of which are with antibodies, anti-HLA antibodies, and they are very often mismatched. And if you look at antibody levels, it's not really sort of indicative how much of the antibodies you have, but how many platelets you find actually back after 1 or 24 hours. It's also not the polyclonality that is sort of indicative of how often this happens, but it's also very important. But if you look at the level of galactosylation of those antibodies in these patients, you see a nice correlation actually. The higher the calculation contents of your antibodies, the less antibodies you actually find back. And I would like to apologize already now because I tend to speak, get very enthusiastic and start very fast. So I was going to speak a little bit longer, but well, I didn't make it, sorry. But I want to summarize now. So basically, I showed you that afucosylated of antibody, they are really good at binding Fc gamma receptor 3. They can sort of shift the balance towards increasing the affinity or effector functions of those antibodies, even sometimes black and white. And I've been obviously using NK cells as a demonstrative purposes, but on myeloid cells that express all the receptors, it's less black and white, but it's definitely there. I didn't go into that. And this is really important for natural immune responses against for enveloped viruses, we think, but definitely in alloimmune responses in pregnancy, where this is very important. Also malaria, and we think that this is somehow the key in understanding malaria immunity is not only to put out new antigens and look at how the immunogenicity is can be enhanced, but we also have to think of the quality of the antibodies. The natural immunity to malaria is really afucosylated response. It's dominated by that, and and those are protective in the endemic region. But somehow, when we do this by vaccination, we have not been able to go so far, at least on the red cell stage. And I think we need to look at this. For dengue virus, similar things are happening, but then these are really pathogenic antibodies. They promote disease because of pathology. Similar things seem to be probably happening in SARS-CoV-2 with this enhanced Fc gamma receptor mediated activity that you see. The virus seems to actually go into these cells and actually start to trigger the replication in these macrophages, but it actually is non-productive for the virus. The virus actually triggers pyroptosis and the cell actually dies, which is also enhancing the inflammation. Other people have shown that. I don't have the references here. But for galactosylation is also very important. So you take the antibodies, you put galators on it. They are much more potent in activating complement. You shift this balance. These are not the pro-inflammatory antibodies, as you see in the literature. It's these guys. These are the guys that activate complement, not those ones. And this is an important thing that we have to take into account in normal immunity, that we are going from this stage to this stage as we age. And this is an important dimension. It's also for immunization. Recently, after immunization, you're also going from this stage. You start with this stage, and in time, you will also go down. But this is something that is important to keep in mind, and I think this is the natural immunity that we have been studying. But basically for you guys, I think for therapeutic antibodies, you can also now pick your flavor in terms of sugars. These are the two sugars that are really matter in terms of functionality. So far we know bisection and silation is less active. It still may do something, and obviously we know of a lot of literature on anti-inflammatory antibody salicylic acid, which seem to be working, but we still don't know exactly how that works. But in terms of enhancing the functionality, these are the two most important flavors. And with that, I would like to thank you for your attention and thank all the past and present PhD students who did this, all our collaborators, especially Manfred Wörer, who has been our eyes and ears basically on the mass spectrometry, and his team, and all the funders, in this case, SonMW and Alispr and Sanken itself. Thank you very much.
What regulates the transferases that do this?
That's a good question. We don't... Yeah, so what regulates the transferases that do this? So we see actually... And people have also looked in the HIV and by I think that was the Galit Aldes group. They looked also on fucosyl transferase 8 expression in these cells. So if you go in and you look at the antigen-specific cells, you actually see that the FUT8 expression is correlating with the amount of fucus that is in there. But what is regulating that expression upstream, we have almost no clue. There are some literature out there that seem to, GWAS studies seem to pinpoint some regions in master gene regulators that are affecting very slight variations within the population, but does not explain the difference in immune responses between individuals. because even similar similar antigens that have similar tendency within a population to cause afucosylated antibodies can be completely the opposite within an individual. So it's not like that some individuals are really good at this and others are not. It's some stochastic thing that is going on there. But we don't know. We're now focusing more on single cell and transcriptomics on those to try to answer those questions. But at the moment, we don't have those answers.
Thank you for your presentation. I'm just curious, when you do the population analysis of the range of galactosylation or afucosylation, do you see whether there are certain donors that have a certain level and have a broader polyclonal galactosyl, say, levels of sugars that are distributed, or do you have people who are just like mostly afucosylated or higher precedence of that, and then they may have a certain range, or is the diversity just all over the place for different people?
The real short answer is the last one. So you answered the question yourself. Now, it does not seem to be related to the amount of bisectin glucanag or anything else. Sometimes we seem to see something that it seems to pops out as significant in one population and one immune response, but then we look at the next, it's gone, and so I don't think there is one. Yeah. Good question.
Hello. For the pregnancy-related conditions, is there a potential viral element to those?
I didn't think about that, but I doubt that. So these are red cell responses, restricted red blood cell responses, and viruses are not really good at propagating in red cells.
Just referring to the propensity for afucosylation more generally in those responses, it could be a correlate with infection during pregnancy?
Yeah, so we've not noticed anything in this. So I understand where the question is coming from. Is there a sort of a pro-inflammatory something else happening that may skew this? I would think that if that would be the case, we would see it to all red blood cell antigens. But some red blood cell antigens seem to be completely immune to this sort of a regulation, and I don't understand that. It's not like we are mimicking, so if anything, we are mimicking malaria infection for the red cell responses, but for platelet responses, that's another story again. And there, it's very similar kind of fucosylation there. So, yeah, I cannot pinpoint anything, but there are, of course, I acknowledge that there are a lot of unknowns.
We'll take one more question. We're just about to go into the lock, so it's going to get very noisy. But,
I have a question. I have a microphone, so I have one quick follow-up question. Are there any natural people that naturally don't have any of the glycosyl transferases, or have people tried this in mice? What happens if you start knocking out these enzymes?
Yeah.
To the immune response?
Yeah, I've heard anecdotal evidence that there is some people that actually don't or have mutations if you take, but I haven't seen any data on that. So and we have never picked up anybody who has is complete futate knockout or something like that. Never seen that. Mice are really peculiar because they have the sensory input or sensor receptor to respond to afucosylated antibodies. Even human antibodies are fucosylated or non-fucosylated. They discriminate that through Fc receptor 4. But we have looked at the immune responses in mice, and they cannot make afucosylated antibodies somehow, at least the inbred strains we are using. Some other experimental animals do, and I think what we need to do is to go into those mice. Yeah, so again, I think that is a very interesting and important question, at least the answer is, because that will also tell us what is wrong with these laboratory mice that we have.
Thanks.
Yeah.
Oh, it's not a quick question, Dennis. No, I will. No, Gestur, just thank you for your story and all these fantastic data, and looking so carefully at all these antibodies at this high level of detail. But just to check my line of thinking, pretty simple. So actually what you're saying, is if IgG gets more and more mature glycosylated, the functionality of the antibodies drops.
No, I would not say that.
They're
So,
less active with respect to complement activation.
yeah, with the level of complement.
Yeah.
Yeah.
And Fc gamma receptor as well with respect to,
Yeah.
yeah. So actually, correct. So, but when the system is in a hurry, infection, you get maybe these far more active antibodies.
So in terms of galactosylated antibodies, early in the
Yeah,
response, and when you make a new response, it's going to be tip top. Yeah.
Tip top. Yeah, perfect. And you also said something about getting older.
Yeah.
And I'm super worried, menopausal woman. So, what should I do? What should I not eat, for instance, to ensure that I have this mature, you could say, completed IgGs?
There is a recent paper by the group of Gordon Lauch, who looked at lifestyle and lifestyle changes and what you can do to actually do that, to change what he calls the glycan age, because there are relationships between the actual age and what you see in the antibodies. And actually, things are going all directions. So it depends on the individual what you should do. So it's I think that I got a little bit lost when I read that paper. But you can induce some changes, but exactly what? I think that's a personalized.
Okay. If folks want to go upstairs, they can watch the dramatic rise of the